In this video, I will describe some of the basic concepts of clinical trial design. Clinical trials start with an intervention and a question of whether the intervention works. The most basic structure of a clinical trial is composed of two groups, one who receives the treatment and another who does not. These two groups are then compared on an outcome of interest. For example, are people who got the treatment less likely to die than the people who are in the control group that did not get the treatment? Our outcome of interest is the dependent variable, and treatment status is the independent variable. Clinical trials may also compare a new treatment to an existing treatment, a treatment to a placebo control, or may evaluate a prevention intervention. Clinical trials may also have more than two groups. In order to start a clinical trial, researchers define who exactly makes it from the population of interest into the study sample using detailed inclusion and exclusion criteria. Potential participants are then recruited and screened for their eligibility based on the inclusion and exclusion criteria. If at any point during the study, the process of including or excluding participants results in differences between the intervention and control group, and these differences are related to the outcome, this is called selection bias. In order to compare the performance of a treatment and control group, researchers need to decide which eligible participants are in the treatment group and which are in the control group. You can imagine different ways of doing this. The researchers could get participants to choose, or the researchers could choose themselves. Either of these methods could potentially create bias, though, because certain types of people might be more likely to select treatment, and researchers might prefer to have certain types of people in the treatment versus the control group. Allocation bias occurs when there is bias in the assignment of participants to research groups. When comparing treatment groups, the goal is to have the groups as equal as possible on everything except for the treatment. This way, if there's a difference between groups in the treatment outcome, we know it's the result of the treatment and not some other difference between the groups. For example, say the allocation process results in a control group that is older on average than the treatment group. In other words, age is related to group assignment. Then say age is also related to the outcome of interest in the clinical trial. If the treatment group has better outcomes than the control group, it would be unclear if the differences were the result of the treatment itself or if the differences were the result of age differences between the groups. Allocation bias is a problem because it creates the potential for confounding by third variables as depicted here. Allocation bias is minimized by randomly assigning participants to the treatment or control group. Random assignment is designed to make the treatment and control groups equal on all potentially confounding variables, so that if a difference is found between groups for the outcome, researchers can be confident the difference is the result of the treatment and not confounding variables. Once the intervention and control has been administered, participants need to be followed over time to see if their health outcomes differ by group. Several things can happen that can result in the loss of participants during this part of the study. When participants drop out of the study or pass away, this is called loss to follow-up or attrition. If there's a relationship between loss to follow-up and group assignment, there's a potential for loss to follow-up or attrition bias. For example, imagine a study that is testing the difference between a new medication and an old medication. Now say that the new medication results in more loss to follow-up than the old medication due to worse side effects. If the people who drop out because of side effects also tend to be the least healthy to start with, then over time, the group receiving the new treatment would become more healthy on average than the old treatment, and it would have nothing to do with benefits of the treatment itself. This would be an example of loss to follow-up bias. Loss of follow-up bias can be minimized by implementing procedures that reduce the extent of loss of follow-up. For example, researchers may have multiple ways of contacting participants or may increase incentives for participating as the study progresses. There are a number of other factors that can create bias while a clinical trial is ongoing, including performance bias, the placebo effect, and the Hawthorne effect. Performance bias results when the performance of the person offering treatment is biased by the knowledge of a participant's group assignment. For example, say there is a study comparing two medications that help people quit smoking. If the physician prescribing the medications knows about the treatment assignment, that physician might try harder to get people to quit smoking if they're taking the medication that the physician thinks will work better. Performance bias can be minimized by blinding or concealing the treatment group allocation of participants to anyone involved in the treatment of the participants. Placebo effect occurs when people's health status changes over the course of a clinical trial because they believe they're getting treated, regardless of whether they're actually getting treated, and if they are getting treated, regardless of whether the treatment actually works. If the placebo effect is stronger in one group than the other, say if a medication is given to a treatment group and nothing is done for the control group, the placebo effect can bias study findings. The placebo effect is countered by giving the control group a placebo. For example, a treatment group may receive an active medication and the control group may receive a sugar pill. Note that this would not actually remove the placebo effect from either group. It would merely allow the researchers to say that if they found a difference between the treatment and control group for the outcome of interest, that it was not due to the placebo effect, because both groups are equally susceptible to the placebo effect. Finally, if there's bias in the measurement of the outcome, this could potentially influence the study results. An example is observer bias, where the person measuring or observing the outcome is aware of the participant's group assignment. The observer might be biased and rate the treatment group more favorably than the control group. Observer bias can be minimized by blinding all of those involved in assessing the treatment outcome to the allocation of the participants. There are a number of types of bias not covered in this video. Some of them are shown here. When designing clinical trials or evaluating proposed trials or evaluating published trials, the important point is to be constantly on the lookout for anything that could potentially bias study findings. Having a broad sense of common types of bias and practicing identifying these types of bias can help train the scientist to critically evaluate and identify potential sources of bias.